Gingiva 3, the microscopic perspective. In the last video, we explored the macroscopic features of the gingiva. Now let us dive into the microscopic features. The gingival architecture is composed of gingival epithelium and connective tissue. This video focuses mainly on the epithelium. The underlying connective tissue will be discussed in upcoming videos. The gingival epithelium can be loosely categorized into at least 3 different types based on their location and composition: the oral epithelium, the sulcular epithelium, and the junctional epithelium. Let's take a closer look at the oral epithelium. It consists of 4 layers: stratum basale, or the basal layer; stratum spinosum, or the prickle cell layer; stratum granulosum, or the granular layer; and stratum corneum, or the corneal layer. Did you know that epithelial cells are structurally mononuclear? They possess intercellular bridges called desmosomes and have a characteristic fibrous protein called cytokeratins. This is why we often refer to epithelial cells as keratinocytes. Starting with the basal layer. The stratum basale is the deepest layer attached to the basement membrane. The cells here are cuboidal to columnar, with a hypochromatic nucleus. They are abundant in cell organelles and undergo frequent mitotic divisions. This layer is also responsible for continuously supplying new keratinocytes, thereby also known as stratum germinatum. Moving on to stratum spinosum. Cells here are larger and polygonal than the basal layer. They exhibit tiny adhesive junctions called desmosomes. Although desmosomes are present as major intercellular adhesions in all layers, the polyhedral shape of the cells and the shrinkage during tissue preparation give them a spiny or prickly appearance. Let's continue. This layer is highly active in protein synthesis, aiding in the production of cytokeratins. Next is the stratum granulosum, named for its granular cell appearance. And why is that? The cells appear granular due to keratohyalin granules. But why do we need keratohyalin granules? These granules are rich in filaggrin proteins, which are responsible for the formation of crosslinks between the cytokeratin filaments, thereby providing strength to the epithelium. Deep, isn't it? As we move towards the surface, the cells appear flatter and wider, with nuclei showing signs of degeneration. This layer also contains the otolint bodies or membrane coating granules. What is so fascinating about otolint bodies? These glycoproteins release contents into the intercellular space, forming a lipid-rich barrier that limits aqueous substance movement. This, combined with the production of other proteins, contributes to the cell envelope's cornification, providing mechanical resistance. Finally, the topmost layer is the stratum corneum. Cells here are larger, flatter, and unable to undergo protein synthesis as their nuclei degenerate. These cells are eventually shed through desquamation. The word desquamation comes from the Latin word desquamare, which means to scrape off scales. Over time, the term became more widely used in medical contexts to describe the natural shedding of skin cells. We've now covered the layers of oral epithelium, but there's more. Besides keratinocytes, we also have non-keratinocytes, or clear cells. Let's explore them. Starting with the melanocytes. These specialized cells produce melanin, responsible for gingival pigmentation. They are found in the basal and spinous layers. Ever heard of gingival vitiligo? It is characterized by white patches on the gingiva. Although the etiology is multifactorial, the absence or insignificant amount of melanocytes is the key feature. Moving to Langerhans cells, these dendritic cells located at the suprabasal layers play a crucial role in the defense. So, what do we mean by that? If you have an infection, these cells will act as your antigen presenting cells. Langerhans cells contain large granules called Burbeck granules. Lastly, we have Merkel cells, specialized sensory cells found at the base of the ret apex. That wraps up the oral epithelium. How intricate is the world of gingival anatomy? Before we move on with the sulcular and junctional epithelium, we need to know about a foundational layer called the basal lamina. This thin layer of extracellular matrix underlies the gingival epithelium. Think of it as the bedrock upon which the gingival tissues are constructed. Basal lamina also helps in cellular interactions, signaling, and nutrient exchange. Next in line is sulcular epithelium. So, how is this different from oral epithelium? Sulcular epithelium is a thin, non-keratinized, stratified squamous epithelium, whereas oral epithelium is a keratinized, stratified squamous epithelium. While the oral epithelium has 4 layers, the sulcular epithelium has just 2: stratum basale and stratum spinosum. Additionally, the melanocytes and Merkel cells are absent in sulcular epithelium. To answer a hypothetical question, can sulcular epithelium keratinize? Yes, under 2 conditions: when exposed to the oral cavity and when the sulcus bacterial flora is completely eradicated. Bamboozled? Wait till you explore the most fascinating junctional epithelium. Without further delay, let us get started. By definition, junctional epithelium is an epithelium comprising single or multiple layers of non-keratinizing cells adhering to the tooth surface at the gingival crevice's base. It consists of a collar-like band of stratified squamous, non-keratinizing epithelium. Visualize junctional epithelium as a lid seal on a leak-proof box. The job of the lid seal is to ensure nothing leaks in or out, just as junctional epithelium seals the tooth, protecting the gum tissues from harmful bacteria. Junctional epithelium is one of a kind. Let us learn why that is so. Junctional epithelium has a very high proliferative rate, which means the cells are regenerated almost every week. Junctional epithelium has completely different layers when compared to oral and sulcular epithelium. Junctional epithelium has 2 layers: an internal basal lamina and an external basal lamina. While the internal basal lamina is formed by the junctional epithelial cells attached to the tooth via hemidesmosomes, the external basal lamina connects the basal layer to the underlying connective tissue. You find a cell to connective tissue hemidesmosome attachment elsewhere in the body, but the cell to calcified structure attachment is unique, which is the beauty of junctional epithelium. Wow! You just finished a brainstorming session on the microscopic features of gingiva, the gingival epithelium. So here is a bonus for you. Go to the description of the video and you will find a link to a highlight chart. Till then, keep it simple, silly.